Next speaker before lunch will be Dr. Teegarden from the Pacific Northwest National Laboratory.
Thanks, Yvonne. It's always a pleasure and a privilege to be in this building and be here. I overheard an interesting comment much earlier in the session today. Somebody said, where's the systems biology? And it occurs to me that it's a great question, and it points out that there's a common misconception that I think many of us are responsible for promoting. And that is that Omics, or these high-throughput assays that we're doing, all these efforts that we're using to generate these data sets that we're going to use to infer something about adverse biology, that is not systems biology, although it's often referred to as systems biology is something else. I think systems biology is more the effort of trying to take that vast set of information, develop some understanding of how these adverse biological events develop over time in dose, and piece them together into some very quantitative, hopefully credible quantitative models we can use for prediction. So there is a big difference between systems biology and what we're really doing now, I think, in the ToxCast-like programs. We're in the information gathering phase right now, I think. So I'm from the Pacific Northwest National Laboratory. This is not an institution that many people know about, so I think probably a very brief introduction of what we do at the laboratory would be helpful for you all to understand the context of my talks and what kind of biases I come with. This is about a 5,000-person DOE national laboratory run by Patel in southeastern Washington. And what's important, I think, to you all is that I come from a group of about 20 toxicologists working in a systems toxicology and exposure science program. And this group of toxicologists covers a lot of different domains, everywhere from systems biology to in vitro toxicology, to in vivo toxicology, a lot of dosimetry and pharmacokinetic modeling, risk assessment, source to outcome modeling, very diverse group of expertise within this. And many of the people come from actually an industry background, so they have experience in the process of product registration and risk assessment itself. But we also have a group of 20 or so people in a bioinformatics and computational biology group led by Katrina Waters, and these are the people who spend all their days and nights working with these large data sets to try to develop models, computational biological models, or just infer patterns from data about the biology that they're studying. They apply that across a lot of different domains, everywhere from environmental sustainability to biomedical sciences or to toxicology, for example. We also have a group of about 60 people, probably more depending on how you count them, that do the omics. In other words, they're involved in proteomics, genomics, lipomics, metabolomics, and I think the laboratory is probably considered one of the world leaders in proteomics. So we are generators of these massive data sets, if you will, across a bunch of different biomedical and toxicological domains, and we are users of those data in the sense that we try to interpret it, and we also try to apply it, those of us in sort of the applied tox field, to real problems where we can. So that's kind of the context that I'm coming to you in. So I was asked to talk about cancer risk assessment and systems biology today, and of course that's a huge area to consider trying to handle in a short session. But what I decided to do in trying to get towards giving you an example, a real-world example, is to try to talk about next-gen risk assessment, some of the opportunities and limitations as I see them, coming to this in somewhat of an outsider in some ways, in the context of a conventional risk assessment for a pair of compounds, which I'll name next. And I'm really going to focus on a couple areas, mode of action mostly, threshold to some extent. And the reason I'm going to do that is because that's where the real difference lies between risk assessment for non-carcinogens and carcinogens. That's what it boils down to, because that's what the dose response depends on, and that's what the eventual regulatory level depends on. And those are the areas that have been terribly controversial, I think, at the level of regulatory agencies, at least in my perspective. And then I'm going to provide some challenge questions, I think. So the two compounds I'm going to talk about are vinyl acetate. I'm going to talk about vinyl acetate, but this is actually, as you'll see, it's metabolized to acetaldehyde. Now, acetaldehyde, I think, is one of the tier 2 compounds on the next-gen example list as I saw it. So it's also a good example for that reason. Now, vinyl acetate is a synthetic organic ester. It's used in the production of resins, polymers, adhesives, acrylic fibers, and a whole variety of other things. It's a high production volume chemical. And if you want to think of it this way, the risk assessment process for vinyl acetate started many years ago, as it typically does in the conventional approach, with a high-dose hazard assessment. That was a blue ribbon or the gold standard 2-year chronic bioassay. And what they observed in those studies after 2 years is that in rats, but not in mice, you got nasal cavity tumors at about 200, at concentrations above 200 parts per million in the rat nasal cavity. Specifically, the olfactory epithelium was the sensitive tissue in the rat, although there was a much lower incidence of tumors in the respiratory epithelium in the nose. These were long latency tumors. They were not, didn't appear early in the assay system and only at very high doses. And what preceded those tumors is olfactory tissue damage. That is, the olfactory tissue tends to be more sensitive, probably for metabolic reasons, and relatively quickly at very high concentrations it's wiped out and it's replaced by a totally different type of epithelium called respiratory epithelium. Along with that, you can see regenerative cell proliferation. So you can kind of see that there's a basic, very common mode of action here for a carcinogen that's really driven potentially by cell proliferation for the most part. Well, that was the beginning, and that really started what ended up being a 20-year, at this point perhaps about $15 million safety assessment program. In other words, you have a high-dose effect. Now you have to figure out what the implications for that are for human health, and that means understanding much more about the biology, much more about the mode of action, and what that means specifically in terms of dose response, in particular, whether or not there's likely to be a threshold. So a whole host of studies were done. There were subchronic studies that were done that were mechanistic in their design to try to tease out some aspect of the adverse effects and what was behind them. Metabolic studies were done. In vitro and in vivo addict studies were done. Mutation dose response in vitro. Pharmacokinetic models were built, both for in vitro systems and the in vivo systems. And, of course, there was a variety of other in vitro mechanistic studies that were done over the course of time. So this has been a massive piece of research to support the safety assessment for this compound. So it gives us some sort of context that we might be able to think about how this next-gen risk assessment approach using principally cells could stack up against this. So the first thing, I shouldn't say it's the first thing, but one important thing that they discovered was that was the metabolic pathway for vinyl acetate, and that was that it was metabolized by carboxyl esterases, which are ubiquitous in the blood and in the tissues. So you get rapid rates of hydrolysis of this acetate to produce acetaldehyde and essentially acetic acid, so protons. So this led to early hypothesis by Matt Bogdanfi and some of his colleagues that it could be, in fact, that intracellular acidification was behind the toxicity that was observed in the olfactory and respiratory epithelium in the rats. And so this was actually confirmed over the course of the next couple years. On the top panel, what you're seeing is preliminary studies that were done in rat hepatocytes, which are metabolically competent for vinyl acetate. And you're looking at a pH shift from normal, about 7.5 on the right side, after exposure to different concentrations of vinyl acetate, the pH dropping very significantly to 6.2. And down on the lower left side there, actually I have a pointer here. These studies were repeated actually in nasal explants. So they took tissue from the olfactory epithelium and respiratory epithelium, put them in cell culture, exposed them to vinyl acetate. Yeah, here. Right, thank you. And discovered the same thing. That in fact the pilot studies were confirmed in these tissue explants that you get significant levels of intracellular acidification. So that was a key to this whole story, and they conducted some subchronic inhalation exposures. This is the data from the 20-day study, and all I want you to see here is that they're looking at cell proliferation, or a proxy for it, at different inhaled vinyl acetate concentrations, and you can see in the upper line here that you see some very significant increases in cell proliferation. This is regenerative cell proliferation in the olfactory epithelium, but much less so in the respiratory, which just doesn't suffer the same way in this system. But you also see very little changes at low concentrations. So environmentally relevant concentrations, you don't see this regenerative cell proliferation, which has some important implications, of course, for the presence of a threshold. But even after all this work, I think that the group understood that even though they had a very strong case, I think, for events that were driven only at high doses, that in fact the lingering issue was whether or not acetaldehyde, which is a known genotoxic material, can form adducts, protein and DNA adducts, whether or not that was contributing, that genotoxic element was contributing significantly to the mode of action. Of course, whether or not that would have implications for low-dose risk assessment. Is it linear or not? And so the research team took up this particular issue. This is work that's done by Les Recio at ILS and Bob Bedinsky at Dow. I should point out that all these studies were funded by a group called the Vinyl Acetate Council, which is a producers association that spends their money trying to understand the toxicology and the safety of this particular material. And I'm a science advisor for that particular council. And what they found when they looked at TK mutations in the mouse lymphoma mutation assay was that, in fact, there appeared to be evidence of a very clear threshold. That is, at nanomolar and below levels, or actually even below low micromolar levels, high micromolar levels of vinyl acetate, there was no change in the mutation rates in these TK mutations. And they took it a little bit further, because obviously that's only looking at one particular type of genotoxicity, and they looked at the standard micronucleus assay, and they found something very similar, and that is that VAM and vinyl acetate, which is here on the left, and acetaldehyde, which is its metabolic product, both show very clear thresholds for formation of micronuclei. And in reality, the increase that you see in the micronuclei here in the blue is really carefully or closely associated with cell death and apoptosis rate. So it's only when the cells are getting extremely stressed that you start to see, and dying, that you start to see the formation of these micronuclei. The other interesting and important thing is that the acetaldehyde curves are superimposable over the vinyl acetate curves. And I think it's been everybody's assumption all along that acetaldehyde is really the driver here, both probably for the cytotoxicity and the genotoxicity. So this is exactly what we would expect. But whether or not that's enough information to convince a regulatory agency that this is actually a threshold carcinogen is still an open question. So the group has begun to work on the issue of measuring adducts formed by acetaldehyde. And I'm not showing you that data here because only the pilot data is available now. What I'm showing you instead is a view of the future for acetaldehyde based on the findings for its cousin endogenous aldehyde, formaldehyde. And this is some very nice work that was, in fact, I would call it pioneering work, because this is going to, I think, very much change the landscape of risk assessment for this class of genotoxic carcinogens, the aldehydes, or at least the endogenous aldehydes. This was done by Jim Swenberg and his colleagues at UNC. And what you're looking at here is some very sophisticated mass spec-based analytical work where they are reporting, in this case, formaldehyde adducts. And these are endogenous adducts produced by formaldehyde that is produced as part of the one-carbon metabolic pathway in the human body. So all your cells in your body produce formaldehyde. And it turns out that there's a very consistent level of endogenously produced DNA adducts, and there's about 4.7 plus or minus 1.8 adducts per 10 to the 7 deoxyguanines. That's an incredible low value. I mean, there's some very sophisticated analytical measures. And what's most important is they developed a method for exposing rats by inhalation at the same concentrations used in the chronic bioassay for formaldehyde to an isotopically labeled formaldehyde. And what you're looking at is the contribution of the exogenous, that is the exposure portion of formaldehyde here, to the total adduct load. So at high concentrations of formaldehyde, this is a carcinogenic dose, you get a significant contribution from the exogenous formaldehyde. But as you move down to even the low carcinogenic dose here, 6 parts per million, only 20% of the total adduct load is actually from the exogenous formaldehyde. As you move down to the lower non-carcinogenic doses, what you find is that these are log orders, 1 and 2 log orders lower than the endogenous. That means it's contributing less than 10 or less than 1% of the total formaldehyde adducts you have in these tissues. That says something really important about the level of cellular exposure in the context of an endogenously produced material, and just something important about how much risk really might be involved in a human population exposed at low levels. So this same data is being put together for acetaldehyde. Now, where this leaves us, in our understanding of the mode of action for vinyl acetate and acetaldehyde, is that we have a material that is metabolized in the olfactory epithelium to two materials principally, protons, which can cause intracellular acidification and cytotoxicity, and acetaldehyde, which in and of itself is also somewhat cytotoxic and also genotoxic. But that those two things, the cytotoxicity, which is surely a driver here, and the genotoxicity, I think there's very compelling evidence now that neither of those two processes are significant contributors at low exposures to this material, certainly at not at doses underneath the carcinogenic levels that we see, exposures that we see. So this has been a hard-fought scientific story. Very careful, specific, hypothesis-driven research over the course of 20 years. Now, this is exactly, in many ways, what we're trying to get around with ToxCast and Tox 21, is to avoid this kind of commitment in funds and time. So then the question is, how do we do that? Or is that even plausible here? So could a systems biology approach contribute to this kind of risk assessment, specifically to the genotoxicity risk assessment? And I guess I give a cautious yes to that, given where we are now. But I think that the platform really has to, in order to do this, it has to be steered truly toward risk assessment. It has to evolve into a platform that really supports dose response assessment and threshold assessment, because those really are the key questions for carcinogen risk assessment. Positive and negative findings for genotoxicity are not going to be enough. They should not be accepted, I think, in this context as evidence that this is clearly a genotoxic carcinogen in human exposures. So what about omics approaches to gene tox? I think there's surely some potential for proteomics and transcriptomics to be used pretty broadly overall to assay biological response to things that actually interact with and damage DNA. And I'll give you just a brief example of that from formaldehyde. Certainly things like adaptomics and epigenomics may offer useful tools as well for doing somewhat the same thing, sort of broad global assessment of, essentially you're peering into the response of the cell to try to get an idea of whether or not in the cell's mind it's under enough of a DNA assault to be problematic in terms of, or a contributor to carcinogenesis. But I think for looking at the problem of threshold, which again this really boils down to, most of the omics approaches just don't have the sensitivity or the quantitative capability to do that. And so I think we're going to have to rely on panels of, for example, PCR probes for key genes from these pathways to provide much more quantitative, much more sensitive data for thresholds, or I'm not confident that they would be accepted without some major changes in how or the expectations of a regulatory agency has for sufficient evidence for proving something is non-genotoxic and mutagenic. Certainly high-throughput enabled conventional gene tox assays will continue to be useful as primary tools or maybe just adjunct tools at a later time. But I think that for any of these tools to really be useful in the future, dosimetry is going to have to become as an important element of the program as response really is now. I mean, all the art, I think, as most of you know, is focused on the response part of the measurement of the dose response. So this is an example from formaldehyde. So how can omics approaches, in this case transcriptomic approaches, be actually used to make a statement about whether or not the material is significantly genotoxic at low doses, or where it's genotoxic might be the right way to think about it. And this is some very nice work that was done by Mel Anderson, Harvey Kluel, and their colleagues. And all I want you to take away from this slide is that what they were able to show in the nasal epithelium after exposure to formaldehyde is that there definitely is a biological transition point, that at 2 parts per million and lower, you really have evidence from transcriptomics or global assay of gene transcripts, cellular stress, thiol transport reduction, and we have inflammation, cell proliferation. All these things were upregulated across all the study, the durations of the study. At higher concentrations, you see a transition where you're actually seeing the cellular response, I believe they believe, to damage to the genome, and some key signaling pathways related to carcinogenesis or promotion, I think. So I think it's clear that these kinds of studies can be used to support understanding of where the biological transitions are on the dose-response curve. And that's a near-term thing. That's something that's in practice now and I think holds a lot of promise. But I think that dosimetry has to, as I said, be a very integral part of this, and the Hamner Group has done a good job of this. And what I would want you to take away from this is it's really time to stop thinking about in vitro systems, as much as we want them to be, as simple systems. It requires a lot of thought, these systems do, in terms of dosimetry. For example, vinyl acetate and acetaldehyde both have important pharmacokinetic characteristics in vitro. This is something that we typically want to ignore. So in our own studies, these are the ones that were done by Bob Ladinsky and the Dow folks and Les Resio, we actually looked at the loss of vinyl acetate in the mutagenicity assay system. And these are just different media, with or without cells, fetal bovine serum and horse serum. And all I want you to see from here is over a span of 240 minutes, which is a relatively short period of time, or less, even 60 minutes, you have complete conversion of vinyl acetate in this case. Okay, virtually complete conversion. Massive loss, production of acetaldehyde. That's the proximate carcinogen. So you have to ask yourself, are you really assaying vinyl acetate or are you assaying acetaldehyde? Acetaldehyde has its own issues because it's volatile. I think other people have pointed out that for in vitro systems, volatile compounds pose a very significant challenge. So if you're losing most of that material or part of that material, you think you're studying millimolar concentrations, but you're not. Okay? These kinds of things have got to be built better, I think, into these ToxCast, TOX21 types of programs. So without dosimetry, the relevance of these tests really cannot be determined. And you have a problem comparing one lab to the other, obviously, and one assay system to the next if you know nothing, or you're basing your ideas on a hunch or the hope that your concentrations are the same across these systems. So, but high-throughput systems or Omics advanced mode of action assessment for vinyl acetate. Could we have done this in the beginning, if we were starting fresh now? And I think the answer to that is I think we could have gone part way. I think there's, for example, if there was some data, structural and chemical data, like the prior speaker talked about, that was able to identify this acetate, these ester groups that could be hydrolyzed to produce acetaldehyde and the protons, that actually is a structural feature that would apply not just to vinyl acetate, but ethyl acetate, propyl acetate, isobutyl acetate, the whole group of acetic volatile esters that are used commercially. So it's certainly the case that that kind of work could identify and flag these as materials that could produce this kind of mode of action. So there's a real possibility of that being an approach that would have worked here. But I think I would feel more comfortable about these kinds of approaches being useful if the appropriate cell lines for the respiratory tract were available. Remember, we showed a very big difference between respiratory and olfactory tissues. So if, in fact, you had a system where you only had some epithelial cell or just the respiratory cells or only the basal cells, then I don't think you'd have a system where you could convincingly you would be convinced that you could capture the right mode of action for this particular class of materials, particularly if they weren't physiologically normal and they weren't metabolically competent. And I think that even though it moves us away from the high throughput kind of thinking, moving toward co-cultures, three-dimensional systems, human on the chip systems, and the use of induced pluripotent stem cells is going to be very helpful here because it will move us toward having, I think, better biological systems, more relevant biological systems to do these studies in. But clearly one of the benefits here is that the omics approaches, the high-throughput approaches, are very unbiased. I mean, I think everybody here should recognize that this group started with a hypothesis and followed it down. And I think after years, I'm convinced we have the right answer and we haven't missed anything significant. But that's always a possibility. And if it wasn't for this particular material, then maybe it would be for propyl acetate or ethyl acetate. In other words, there is a value to that global assessment that you get when you use those approaches. So, some limitations, at least in my perspective, and I hope I carry the perspective with me of some of my colleagues. Multiple mode of limitations, if you will, of omics-based approaches for mode of action. Multiple modes of action. In other words, it's been my experience in the few times that I've worked with these data, and I think the experience of my colleagues who have done it more often, that there is rarely a single story that comes out from a massive transcriptomics database, even for one material, or two, or three that you're trying to work on and compare. And I think somebody else alluded to this. If you know the answer, you can look at these massive data sets and find the information in there to support that particular answer. But if you don't have that, this is a fresh compound, no modes of action, I'm almost certain you will always be in the case of having 10 or 15 modes of action or 3 or 4 compelling stories. And I think that's going to continue to be a challenge for this field. Uninterpretable misleading cellular processes, categorization information from KEGG, GO, and Ingenuity. Others here probably know more about this, but I think the point is that these are curated databases, and they're based on our current status of knowledge. People have made decisions on how to bin these things. So the answer you get when you apply these depends on that, and I'm not convinced that they're always right. I think that can be problematic, and I'll show you some examples of that. Limited coverage of cell type, absence of necessary cell-cell interactions. That's one of the big gorillas in the room, I think, that doesn't get addressed, is most of this high-throughput stuff is done in non-intact animals. So you're not getting the cell-cell interactions that are required for a number of modes of action, and that's always going to be a limitation until we move to things like the human-on-a-chip systems that people are talking about developing now. Time dependencies, no need to talk about that. Others have brought it up. So another one, and I'll give you an example of this, is that different answers, you get different answers from metabolomics, transcriptomics, proteomics, and any other that you want to apply. So as we move towards the ominome, all omics, and integrated omics, you're going to have 4 answers for one material, 3 modes of action from the metabolomics that are suggested, a couple from the transcriptomics. Try to integrate them all, which is something that Katrina Waters' group does, and you're probably going to get another answer. Okay, so let me try to give you some examples of this. So where's the carcinogen? Okay, so let's look at this data set. These are processes that were common to, it says common to A, B, and C, and A is the low-dose group, B is the mid-dose group, and C is the high-dose group. So we binned processes that were common across these doses for this particular material. And they're placed here for you to look at. And all of you should be looking, and you should see at least the word cancer many, many times. Okay. In contrast, this particular material, if you look carefully at this list, I hope you can read it, have some processes working. You have things like microtubule skeletal organization, mitotic cystic chromatid exchange, xenobiotic response. So maybe some of these suggest carcinogenic pathways. But you've got things like cellular carbohydrate.
Come back to the mic.
Oh, sorry. You've got things like cellular carbohydrate metabolism, which really tell you very little about what's going on. So if you had to decide which one of these two was a carcinogen, I'm probably betting most of you will say this is a carcinogen. This one is absolutely not a carcinogen. This is amorphic silica. It's an inflammatory compound. The inflammation resolves after inhalation fairly quickly after a couple of days, whereas this is mixtures of PAHs. Okay? And there were different approaches that were used in these two examples, but the point is I think that there's always going to be some trouble in interpreting these things unless you kind of a priori know the answer. So here's another one. Is this a genotoxic carcinogen? And if you look carefully at this list, you'll see lots of inflammation signaling going on here. Lots of it. But there's also a few other things going on. And you can see, most importantly, probably down here, triggering of DNA damage. Now, these are, I think, GO categories. So cell cycle arrest, response to hypoxia, oxidative stress. And I guess the question I would pose is, if this was the data set you had and a regulatory agency was looking at it, would this be enough to trigger a flag that says this is a genotoxic carcinogen or not? Because with these massive data sets, the likelihood of finding something like this is always there. It's lurking. So it turns out that this is not, I think, a genotoxic carcinogen. This is just endotoxin, lipopolysaccharide. And to make things even more complicated, it is not a genotoxic carcinogen. It's just a classical, extremely strong inflammagen. But, of course, inflammation itself can be a promoter of carcinogenesis. So now the question of whether or not this is a carcinogen becomes even murkier. So what about integrated omics? I hinted at this before, and the treatment here doesn't matter. What matters here is that we're looking at categories of pathways and processes that were stimulated by this particular treatment. The blue lines are microarray, the red is proteomics, and this yellow group is looking at protein phosphorylation, so cell signaling. And what you can see here is, although there is some overlap, each of these three different methodologies implies that different processes are the dominant processes. So again, as we start to integrate these, are we going to have better knowledge, or are we going to have a more confusing, complicated data set? I don't think there's an answer to that. And there's probably not one answer to that. It's just not that simple. So I wanted to make a statement about, and maybe this gets to the systems biology question, to the pathway modeling that's been proposed for dose response assessment. Now, this is just a pathway that comes up from global transcriptomics analysis of response in epithelial cells treated with amorphous silica in cell culture. And the only point there is it's immensely complicated. Okay? There's some key information in there, but about the pathways that are involved that I think could be turned into pathway models that potentially could be quantitated over time. But I want to point out that I and others have worked for many years on the development of PBPK models for use in risk assessment. What's interesting about the difference between these two is that for PBPK models, we can go into the laboratory and one way or the other measure every parameter that we need for these models relatively easily. And for the most part, they're not terribly dynamic. You have issues with enzyme induction, changes in metabolism over time, which can be important. But this system is not particularly dynamic. We can reach out and collect all the data that we need, and still we struggle to fully develop these to the degree that they're acceptable to a regulatory agency. Okay? That's been the case. This is a highly dynamic system in which we have almost no ability to reach in and measure the states of most or all of these variables over the course of dose and time. That's just an impossible notion I would submit to you. And we've been through this process. I'm getting low on time, so I don't want to spend a lot of time here, but we've done some computational modeling of these of pathways of response to silica, and we've done them by developing ordinary differential equation-based approaches that actually describe the evolution of gene transcription over dose and over time, which is a real challenge. And what I'm showing you here is that we've picked out groups of genes that are co-regulated, and we've taken out, we call those clusters, we've taken a particular cluster out and developed one of these computational ODE-based models for this particular cluster. And when considering how to actually model that, it became very clear that we just couldn't do that. The system itself is far too complicated. So Michelle Archuleta spent some time using in silico methods to actually knock out each of the regulators that were involved in the gene expression for this cluster. And by sequentially knocking them out in silico, each of these particular drivers of gene transcription, we're able to come up with a much simplified model that drives, so really only one particular protein drives the expression of this cluster of genes, which we thought would be a much better approach to actually conducting dose response assessment. The ultimate goal actually was to develop a no effect level based on an in vitro study, and I'm kind of zipping through this because the punchline just is that after all that effort, which was not insignificant, we were stuck with a linear extrapolation, which is not at all where we wanted to be. And it's just to point out that sometimes it's a little bit bedeviling working with complicated systems like this, and I am not a person that believes that creating wiring diagrams, that is sort of the essence of systems biology, is going to be a near-term approach to doing dose response assessment for risk assessment. So, final point, and I just can't leave without making this point. Because really my thread in my professional life is dosimetry. And that is that I would make a case to you that there's no toxicology without dosimetry. Maybe that seems obvious to you, but I think everybody here that's involved in the systems biology and systems toxicology will acknowledge that for a long time, even before this field came up, almost all the art in toxicology and lot in biomedical sciences really is focused on the y-axis, response. All the omics, all the proteomics, genomics, transcriptomics, you name it, high-throughput assays, the focus is on how do we generate better and larger amounts of y-axis data. And I would argue to you that that data is only as useful as the credibility of the x-axis data you have, which is the dose data. Okay? That is, if we're measuring things in these in vitro systems and we have no real sense of dose, or we don't have any sense, Dean Jones is going to talk much more about this, about what human exposure levels are, and use that information about human exposure levels to inform how we do these in vitro systems, then we're going to be in the position of just not knowing where any of this falls on dose response, on the dose response curve for humans, for rodents, and no ability to translate that data from in vitro systems to in vivo systems. In the field of nanotoxicology, there's been some very notable papers that have failed to show in vitro to in vivo extrapolation. I think we now have one of the first examples of being able to do that, but only when you deal with the dosimetry issue for the in vitro assay and the dosimetry issue for the in vivo system. So I think we got to get around this particular problem, and I would hope that in the future, in these systems and programs you're developing, that this gets as much attention as the y-axis, because the y-axis is really getting lonely. And just to make a final example of this, bisphenol A, many of you know this compound, it's incredibly controversial. But one of the reasons it's so controversial, in my opinion, is that there, up until this point, recently, there really has not been a systematic effort to put into context, that is the human exposure context, all the rodent studies that have been done and all the in vitro studies that have been done in terms of dosimetry. In other words, is the dose of the bioactive form of the material in the in vitro systems and the rodent test systems anywhere near what human exposure is? Okay? And the answer to that question, when I look at the dose problem for humans by 5 different methods, I get the same answer by all of them. There's a convergence. And the answer is that human exposures to the bioactive form, that is the blood concentrations of this material, are on average in the picomolar range or the sub-picomolar range. And even at the 95th percentiles, they're not a whole lot higher than that. But all the concentrations, blood concentrations, where you see effects in animals and in cell culture systems are really orders of magnitude higher. So, what does this tell you about if you're studying an endocrine active compound like this in an in vitro system and you're not thinking about human exposure? You might come up with this being a very important endocrine disrupting compound, but you'll find out you've just studied it in a dose range that didn't matter at all. So dosimetry really matters. Human exposure science really matters. That's where the future here is, I think, and Dean, I think, is going to follow up and build on that. So I am out of time. Challenge questions you can read. I'm trying to think if there's any I really want to say before I walk out of here. I think you can read them, and maybe I'll just open the floor for questions instead of running through these.
Thank you. So I'd like to invite the folks who are listening online, and I've been informed that we have very vigorous participation, over 250 people listening to the webcast, so please submit your questions. And for those asking from the microphone, please try to ask a short question, and a short answer would be appreciated.
Sorry, Marie from USCPA. I have a couple of comments. One thing regarding the omics data. And a lot of times what happens is like you have data generated, like say from genomics in one lab, proteomics in one lab, metabolomics in one lab. We try to integrate the data and see the commonality. But often we may have a problem in terms of having the same set of in vitro system or in vivo system, or the dose, time of exposure, and all those things. I think it would be very nice if one can generate data simultaneously using the same, either the in vitro or the in vivo system, with all the omics approaches, so that we have a better, meaningful integration of the data.
That's a great point, because these problems are complicated enough without those particular confounders in your data sets. And to give you an example, in our nanotoxicology program, we did just that, in that we did an in vivo inhalation study to biopsy mice for 4 hours. And we took the lung tissue for genomics. And in vitro, we did a 4-hour exposure to an epithelial cell type, which was a target tissue in the target cell in the lung. So it's the same hours of exposure, the relevant cell type. And when we did that and properly dealt with the dosimetry, because we measured the dose in the in vivo system and the in vitro system, it turns out that the inflammation, the doses on a target cell dose that cause inflammation in vitro were the same as those that cause the inflammatory response that you can observe by genomics in vivo. But without that careful coordination, your odds of actually seeing these kinds of correlations, I think, are pretty slim. Yeah.
Okay, the other one I have is regarding the endogenous adducts you have shown with formaldehyde. That's a very elegant experiment showing like with the C13 label, the monoadducts of formaldehyde. Those experiments are really great, and they address the systemic delivery of formaldehyde. It shows that it cannot go beyond the point of entry. But it does not address the systemic effects of formaldehyde. And the other thing is, those adducts are more used like as a biomarker, these monoadducts, they are not as mutagenic or mutagenic compared to the DNA protein crosslinks. Most of the time, the DNA protein crosslinks, which have been correlated with the tumors in the nasal passages of these rats. And so this is not only a problem with formaldehyde. You have with ethylene oxide, you have with vinyl acetate. These endogenous adducts, their levels are very high, but they are also repaired very fast. So we need to look at them only as a biomarker. Probably it's probably not so much justified in terms of how you can arrive at a risk with them. So just a comment on that.
Yeah, so I'll avoid the normal debate over the nasal tissue bit, but I'm going to disagree with you because you're certainly right that we can use them very well as proxies for exposure, cellular exposure. I'll disagree with you that you can't use them as a measure of genotoxicity because I think you are actually measuring access quantitatively of a mutagenic compounds to DNA, and you're not looking at every single adduct. But to the extent that the other adducts follow, and I think it's not unreasonable to suggest that they do, you are measuring, I think, a good proxy for generally genotoxicity for these particular compounds. And most compelling is it goes far below the endogenous levels. And to me, regardless of whether you're measuring the most mutagenic adduct for formaldehyde, it's still telling you the same thing. It's telling you that you are far below the endogenous contributions, which presumably would be all the same, right? The same spectrum of adducts would be from both. So we'll just have to disagree there and see where the science goes in the next couple of years. Good question. And great points that I glossed over, by the way.
Thank you, Justin. So let's maybe get back to the systems biology risk assessment and away from formaldehyde. So, Dan.
Dan Krusky, University of Ottawa and Risk Sciences International. Terrific presentation. Lots of really detailed information on the molecular events involved in the carcinogenicity of the agents that you presented. I'm not sure where you come out, though, on how optimistic you are about charting pathways of toxicity pathways underlying carcinogenesis. I'm a pathological optimist, and we're currently doing a project with the International Agency for Research on Cancer looking at the 107 known human carcinogens recently laid out in Volume 100 of the IARC monographs. And we're going through and extract the information on each of those agents with respect to 28 different fundamental biological changes that could be involved in carcinogenesis, observed either directly in human studies or in vitro studies involving human cells and cell lines. Doing the same thing for animal studies, both in vivo and in vitro. Also looking at the 11 hallmarks of cancer that have recently been charted. And my optimistic self is hoping that after we go through all of the known human carcinogens with that level of detail, which is basically examining every shred of information on mechanisms of human cancer, we might get some clues as to what's going on. So
so you caught me, yes, appropriately. You've called me on it. But I'd make the point that the two approaches that we're taking are very different. I looked at it very narrowly in the short time I had time to prepare for this meeting and in the context of these two materials, not sort of globally. I have a diminished optimism compared to yours, I think, but the most optimism that we will be able to use omics and high-throughput methods to actually separate out, for example, genotoxic and non-genotoxic compounds as classes, and that we'll be able to assign materials to various modes of action in general. I think that's very doable. In fact, that's probably the most doable part of this whole thing. As we go down to developing models of systems biology model, wiring diagrams, bioengineering models of cell processes, and using them for dose response credibly, my optimism is far, far less, at least looking out 1 or 2 decades. Okay? But I share some of your optimism for that part. In the middle here, whether or not the kinds of omic technologies and that massive data sets are actually going to be able to provide us with enough information to convince a regulatory agency that, for example, there's no genotoxicity or that this particular material is not a problem, especially given limitations of the number. I made this point before. I think our coverage of cell types is a problem. Our coverage of cell interactions are a major problem. So those are my concerns. And you've thought about this more deeply than I, and you probably share some of those.
So maybe if we average our OQs, our optimism quotients, we might be in the neighborhood of the truth. Great presentation. Thank you.
Thanks for the question.
Isla?
That was a very nice presentation. Thank you. So one of the things I wanted to was as much a comment and then to elicit your response to that. But listening across a couple of different talks and listening at the Human Variability Workshop that was the last NES workshop, I hear two different concepts, and I also observe that I don't think the groups use the sort of same language or constructs, and so that excuse me, that people tend to talk across each other. And one of the things your data beautifully illustrates is the threshold effect seen for these carcinogens. But I think that in the minds of people who tend to be more epidemiologists, I would say, or population biologists, is what they would tend to say is that while individuals clearly have thresholds, and rodents who tend to act like an individual because they're not very diverse tend to have thresholds. If you begin to incorporate population variability, at a minimum, as you make that variability distribution wider, you tend to linearize or flatten out the dose response. And so that the observation of a threshold in the animal data doesn't necessarily translate into a threshold in the population data. And I have to self-identify as an experimental toxicologist like yourself, so I'm most comfortable with the experimental data. But I do know from working in the criteria pollutant program is that when you look at the few very large human population data sets, you see in the experimental evidence, you see the same kind of threshold effects that you've illustrated. But when you look at the epipopulation, you tend not to see them. And I think there are a variety of factors that tend to obscure any population thresholds that may exist. So, one of the things that has to happen as we move forward, I think, is these two communities need to talk. I think experimentalists like ourselves need to incorporate more of this notion of human variability. But can you see a way that actually helps these two groups that have very different concepts on threshold sort of talk to each other?
Well, you're asking me to solve a problem you haven't, and I have 10 seconds to come up with the answer.
Like the last person to say, we'll put that off till.
That's
yeah,
right. Thank you for your comment, Isla.
I think we always get in a pickle when we use the word threshold in general, and I think part of the point, you see these things in cell culture systems too. You see variability when you look at 100,000 cells and who's responding and who's not. So there's populations everywhere. And I think the other way to think about it is not talk so much about thresholds, but when the slope of the dose response curve becomes relatively insignificant, even in that left-shifted population, maybe we're just talking about a level of risk that we're not going to regulate to or we're not of concern. We get hung up in threshold, zero response. And maybe that place is hard to define, but places where there's almost no slope in the dose response, that's a different thing. Maybe that's one way to approach it.
Justin, let me take maybe this question here, number 5, and ask you in the context of your presentation. So there's clearly point of departure, and should you just be looking for cytotoxicity and DNA damage for vinyl acetate or acetaldehyde or something else, and you don't know about that low dose or you don't know anything about the exposure levels, you would probably identify that as a hazard in some screening model, right? So what would you do then, should you're doing this, screening for hazard, and you're not necessarily have a good idea about the exposure levels and everything else, how would you deal with vinyl acetate should it be chemical X?
So you're asking me to answer the question in the absence of the knowledge I need to answer the question, I think, which is kind of the challenge.
But this is,
you're right. It would pop up in a hazard assessment because presumably in vitro systems you do a broad dose response. You would in hepatocytes or something, metabolically competent cells, you'd see the cytotoxicity. If you're looking, you'd see the pH change. So you'd flag it as something highly cytotoxic, possibly genotoxic. So the question then is, if you happen to know, because whatever you're omniscient, that this one is not really going to be a carcinogen at human exposure levels, right? How would you address that in this particular program?
And can you derive new defaults?
Without any knowledge of human exposure. I don't have.
But you'd
an
have
answer
to then
because
label it as potentially hazardous.
you would have to label it as hazard, but again, I was trying to speak to the risk assessment question. It'll get flagged, but then it comes down to how you apply these particular high-throughput and omics approaches to get at the question of dose response in relation to human exposure assessment, which is going to be the next part, because I do think that's critical to put it in that context.
Thank you.
Have I dodged that question appropriately?
No, that's fine. Let's thank Justin for his wonderful presentation.